Great, thanks for the invitation to come and speak with you today. The work I'll be sharing with you is from multidisciplinary collaboration between myself, who's the engineer lead in the projects, and Noah Fearer, who's the lead microbiocologist in these projects. I'll share with you briefly 4 studies that we did together. One was a 1,200 US home study. It's a cross-sectional study where we visited, in theory, by mailing items to the homes once. We did 15 Boulder, Colorado homes, which was a subset of the 1,200 homes. And this was a longitudinal study where we looked at each home for over a year. We visited CU Boulder dorm rooms with sampling between 2.5 to 12 months. And then the Boulder, Colorado flood study where we were able to sample for 1 month. The take home from the 1,200 home study is that if you want to change the types of fungi you are exposed to in your home, then it is best to move to a different home and preferably far away. If you want to change your bacterial exposures, then you just have to change who you live with in your home. So let's dig more into this. We mailed sampling kits to each home that enrolled in the study through the citizen science project, Your Wildlife in Your Home project. The dots on this map are all the participants in the study. We had each participant swab the outer door trim of their home and the inner door trim of their home, assuming that this was a place that was rarely cleaned in the home and that collected dust throughout the course of the people living there. This figure shows the differences in observed richness for the bacterial and fungal communities. Indoor communities were on average distinct from those found outside. And the indoor communities had greater diversity. First, many of the outdoor communities were dominated by only a handful of microbial taxa, while indoor communities had greater Shannon diversity. Second, the bacterial and fungal communities found inside the homes were more diverse because they included both outdoor and indoor taxa. They were also very distinct microbial communities. First, many of the outdoor communities were dominated by a handful of microbial taxa. Oops, we already talked about that. These differences were larger for bacteria than fungi. In part, certain taxa were relatively more abundant and prevalent indoors than outdoors. We found these fungi were more common inside the homes, and only about 1.1% of taxa were relatively more abundant inside, so a very small part of the taxa. But out of that taxa, we found that there were more household molds, more wood degraders, and more human skin-associated fungi. Inside, only 1.6% of the bacterial phylotypes were significantly more abundant indoors than outside. And these included bacteria from human skin, so that's the top bar where we see the purple being more indoors, and then also feces associated, so that was also more indoors. As well, we found that the number of inhabitants and the male to female ratio were significant predictors. Now I want to move to the geographical distribution of our samples. Let's briefly explore these maps, and on the top we have fungi, and on the bottom we have bacteria. We found that outdoor environment strongly influences the indoor fungi. And this was predictable with variables including soil, climate, and geography. We found these broad regional patterns where we see homes in the eastern US have distinct communities compared to those in the west. These distinct differences are partially driven by homes in the eastern have relative abundance of Basidiomyota, and the western homes have Asomyota. That's how the engineer says it. The home occupants structure the indoor microbial communities, and these are not predictable by parameters including soil, climate, and geography. And you can see there's no real distinct geographical pattern here. I'll share now briefly about the 15 Boulder, Colorado home study. These 15 homes also participated in the 1,200 home study we just discussed. But what we wanted to explore was this idea of whether we could go into a home just once and be representative of what's in the home typically. So we designed this study to come into the home multiple times over the course of a year. And the takeaway from this study is that most airborne bacterial communities' homes are just as different a few weeks apart as they are many months apart. We sampled indoors and outdoors with active samplers, and that was collecting all of the air through a filter, and so we have no size cut for the particles. We visited 15 homes, 2 days per season, and 4 seasons for a total of 120 samples. This figure shows relative abundance of taxa for the 2 end member homes in terms of variability. The one on the left was the most variable home, and the one on the right was the least variable home. We see that specific to indoor air, we saw Microbacterium and Pasteurellales. In this figure, we wanted to look at how the abundances might be interrelated to the dissimilarity of the communities in the home. So the top graph is showing that the most temperable variable communities had more stable, lower average microbial loads. So we can see that in which we have a lower value means the home has the same composition across all samples, and a value closer to 1 means the home does not share species across samples. The lower figure shows the degree of variability differences across homes, with home K as the most variable and home Q as the least variable. And I think in here I'm home C, in case anyone's wondering. But what was interesting about this was similar average variability we found was between indoor and outdoor air, with the same average dissimilarity of 0.8. So this highlights that both indoor and outdoor airborne bacterial communities changed substantially over time in this study. And now to the CU Boulder dorm rooms. The take-home from this study was that by examining the abundances of bacterial genera, we could predict the sex of the room occupants. And this hypothesis came out of the 1,200 home study, where we had the ability to really understand communities by the number of inhabitants and the male to female ratio in the home. But we never had any homes in that study, unbelievably, that only had women in the home. So here in the dorm, this dorm at CU does have just men in one room and women in the other room. So we passively sampled the indoor dust in these homes for almost 3 months. And then we also looked at the fan coil filter that was in the room, and that recirculated air in the room over the course of a year. What we found was that the qPCR-based estimates were not significantly correlated with any of the measured factors except for sex of the room occupants. We found that bacteria were in general 67% more abundant in male versus female documented rooms, occupied rooms. And the taxa that were associated with these higher bacterial loadings were skin and feces associated. So here is showing those taxa. On the left, we have the lactobacillus, and that was much higher for the female rooms compared to the male rooms, whereas the Coronabacterium was higher in the male rooms, as well as skin-associated bacteria. And finally, I want to share with you some of the work we did during the Boulder, Colorado flood. The take-home of that was that the impact of flooding is persistent. Bacterial and fungal community composition and fungal abundances are different in flooded homes despite remediation. We only had the chance to visit these homes 3 to 4 months after the flood. They had all been remediated, but it would be very interesting to have a study where you could go back a year and 2 years later. In the study, 7 of 15 homes were flooded. And this is a great picture of just how devastating this flood was. We're still rebuilding, and we just had a major road through the mountains just finished this year. We sampled 50 homes. About 2/3 of those were flooded, and 1/3 of them were not flooded. And the interesting thing about this flood was that it did not uniformly affect all neighborhoods. So the red homes were not flooded, and the blue homes were flooded. And most of the flooding came from groundwater intrusion. A few homes had sewage intrusion, and a few actually creek water was running into their basements. The homes in the study could enroll if they all had a below ground room. We did not want a home with a crawl space, but yet a basement, either finished or unfinished. And we came into the home about 2 months post-flood. All homes had already been remediated or were almost finished with remediation, and we sampled with our passive samplers for 1 month. This figure compares bacterial and fungal communities in basement air of flood-damaged and non-flood damaged homes, and shows that they are statistically significantly different. The abundances were also different in the flooded homes, but it was only significant for the fungi. And we found that in the flooded homes, they were 210% higher compared to the not flooded homes. And the flooded homes were about 76% higher, but it wasn't statistically significant. So that's all I'm going to share with you today. I really want to thank my colleagues who helped pull all this work off, and the funding agencies. And these are the references for these studies. Thanks.